Hi! Today I am here to inform you on what optogenetics is, how it works, the types of improvements it has had on science, and my opinion about it. What is this technology? Optogenetics is a technology that is used to control neurons' activity in the brain using genetic engineering and light. This is done with neurons in the brain, and it essentially controls the brain using light. To help define things a little bit further, genetic engineering is when a living thing's genetic codes are changed and transformed. Once this has occurred, scientists then use light to see the different effects that it has on the neuron. How does this technology work? In optogenetics, scientists add a new genetic code to the neuron they would like to study. When these new codes are added, it allows the neurons to make proteins called opsins, which are responsive to light. Once these opsins have been illuminated with light of the correct frequency, the opsin undergoes the transformational change. It leads to the channel opening or pump activation, cell depolarization or hyperpolarization, and neural activation or silencing. Since optogenetics has been discovered, various opsins have been engineered or discovered, making it possible to stimulate or inhibit neuron activity and discover intracellular signaling pathways on a fast or slow scale when exposed to different length waves. Optogenetics has allowed for scientists to understand the way the neural circuits act and the idea of natural behavior. They've been able to control these neurons with the use of light and digging deeper into how the brain is made up. In 2003, scientists found that channelrhodopsin, found in algae, responded to light by opening and closing. In 2004, this gene was inserted into neurons. When they turn on the light, these neurons are exposed to the light, therefore turning the neurons on and off to see how they respond to light. Through the adoption and adaptation of optogenetics, it has enhanced the understanding of neural circuit dysfunction that underlie such as mood disorders, Parkinson's disease, and addiction. In newer research found, scientists can inject different proteins that affect the speed of the neuron when exposed to light. This has allowed them to be able to turn off certain neurons within a neural circuit. In 1979, Francis Crick illustrated how he believed the biggest challenge that neuroscientists were facing was the control of one part of the brain, specifically a brain cell, while another part of the brain could be left untouched and unchanged. We have come very far that we have no longer needed to worry about harming other parts of the brain, as neurons can be left alone. At the time of Crick's explanation, he understood that light could potentially turn neurons on and off, but he did not know at the time what exactly that would require. Shedding light on the brain's secrets is an extremely compelling article as it goes from history to now and shows the challenges that were faced along discovering optogenetics. Most tests were done on mice as they have similar brain makeup as humans, and humans did not need to undergo these trial periods of testing out these proteins that were going to be put into the neurons. There are hundreds of large and complex neurons within the brain, and different types of cells were compatible with mice that were in humans. That is one of the reasons they decided to use animals that had a makeup of just like humans instead of using humans. I agree with optogenetics. It has allowed scientists to understand the makeup of behavior in more depth. Much like any technology, there are drawbacks to using it, such as possible long-term harm or short-term effects that may be harmful too. Fortunately, with optogenetics, there is a lot of control when conducting experiments, especially using light, as it can simply be turned on and off. Scientists can pick which neuron they want to target, as one protein is usually inserted into one neuron. That way they have control over that one specific area. They can see the impact that it would have on the single neuron as well. I believe this technology has contributed to a lot of science and the understanding of human activity in the way that we behave. I believe that it is a very big contribution to the healthcare system, and I urge scientists to keep using this technology. I hope that I have been able to give you a little bit more insight on what optogenetics is, how it works, and how it benefits the healthcare system. It has become such a big part of the healthcare system, especially being able to alter brain chemistry essentially and be able to find out what and how different disorders have come about. Being able to compare an older brain to a newer brain, a normal brain to a disformed brain. Optogenetics has allowed us to explore all of these different areas of the brain. It has also been minimally invasive as well. It's done with the injection of proteins rather than massive surgeries. It is done with light being able to be turned on and off. It is one of the less harmful technologies on the market, and I really do hope it becomes an evolution.